is Julian Esselborn. Julian got his PhD in chemistry from Universität Ruhr Bochum in Germany, and is working with Kiftesh Khan in both Theme 1 and Theme 2 directions to experimentally validate synthesized metallofolds and to combine them to design a protein assembly with multiple redox cofactors. And he's going to tell us more about that in more detail. Thanks.
All right. Thank you. Thanks for having me, and good, well, pre-noon from the West Coast. And yeah, I'm going to talk a little bit about mimicking metalloprotein evolution by combination of metal binding motifs, what I called it. I also do want to talk a little bit about what the Tejan lab is doing, because I think we're a little bit further away from everyone else. So yeah, I wanted to give you a brief idea of what Tejan lab is doing, and of course also what I'm doing, but that's going to be after. So there's a whole bunch of topics we're working on, going from something like a combination of hydrogels and protein crystals, which is more towards a little bit material science. But then also what I'm going to talk about today mostly is a little bit about nitrogenase, and then mostly about this idea of having methods at protein interfaces, which has quite an evolutionary component to it, and how can we build protein quaternary assemblies, maybe with function and metalloprotein assemblies from that principle. And I'm just going to briefly talk about nitrogenase. I don't think I have to mention too much of the basics, but definitely it's a crucial enzyme for life on Earth, and it's one of these multi-protein, multi-domain machines that this whole group is actually in a way interested in. And it is made from this iron protein homodimer on top in blue, which uses ATP as energy input basically, and then shuttles electrons from this actually interfacial 4-iron phosphor cluster all the way down to the iron MoCo cofactor in the active site. And as a relay, it has in the middle not a standard 4-iron phosphor cluster, but this P cluster, which is basically, if you want a combination between two 4-iron phosphor clusters sharing one sulfur. And this is what we've been more interested in the past, or more recently. And actually, Hannah Rutledge is the fourth year PhD student working on this topic. And so she and of course, Akif are real experts in nitrogenase. And what has been shown by our group before is that this P cluster, if you reduce it by 2 electrons chemically, at least the one from Azotobacter vanillandri is actually changing its structure quite a bit, and it does coordinate now to a serine, to another oxoligand in the back, and over here, and also to an amidate from the backbone over here. And this oxoligand changes depending on species, so it can also be a tyrosine down here somewhere. And so what Hannah did is she did, well, a lot of biology on Azotobacter. It's not that easy, but was able to produce a number of variants of the Azotobacter nitrogenase, which basically either take away any of the oxoligans or put in 2 oxoligans, or swap basically the Azotobacter one for the gluconoacetobacter one, which would be the tyrosine. And so she measured a bunch of activities, and it turns out that de-swapping doesn't do very much to the standard nitrogen reduction activity. However, if you look at the crystal structure and this is the variant which doesn't have any of these 2 oxoligans, and if you look at the crystal structure of the reduced state of, the oxidized state of this variant, then you see that suddenly some of the ions are missing. And so that tells you 2 things, I think, which are important also to keep in mind when always thinking about these clusters and evolution. One of the things is that these clusters are much more structurally dynamic than we normally think they are, I think. And this is, of course, not the only example, but recently, we've seen that for FeMoCo, we've seen that for some of the nickel iron hydrogenase accessory clusters and so on, that there's a little bit more sometimes than what you see at first glance. And it also tells you that some of these additional primary sphere ligands that you don't really pay too much attention to in the first place, they might be a little more important than what we think they are, and we should pay attention to that. Okay, going back to the big picture of nitrogenase, well, you all know yeah, looks a little bit like I did this with Ariel. It's not. So you can just take it apart basically, and it turns out you have small modules, of course, which in this case are mostly Rossmann folds. We all know that they come together then to form folds or domains, which are important for recognition, catalysis. But really, nitrogenase is also a prime example for the supramolecular quaternary assembly, which gives it really the intricacy of the function and the diversity and the sophistication and how this whole works together. And so this is really an interesting, important topic for us is how do we get to supramolecular quaternary assemblies? And the core of what the Tejan lab is doing is this new technique that basically the Akiv kind of invented, which is metal-guided protein assembly. And the idea is something that is, well, I guess in an evolutionary context, it also makes a lot of sense. So I think it's a really important idea. If you have a protein which is a monomer and doesn't interact with the other proteins, and you just install a few metal binding residues on the surface, and this can be something as simple as a Bissier clamp, right? Which can be 1 or 2 or even just 1 mutation away from what you already have, given that a lot of glutamates or histidines are on the surface anyways. Then a lot of time, if you just add a metal, that is enough to form something like an oligomer with metal coordination shared between 2 of or 3 of the monomers, right? And this might be relatively transient, but it mostly is stable enough to at least crystallize the protein in that state. And once we have that crystal structure, it's a great starting place because it's kind of a proven interface that already exists, and now we can improve, right? Using either computational, so Rosetta design software, or just by basically rationally deciding which amino acids could be good. And from there, you can improve it in a way that at some point, maybe you don't even need the metal anymore, right? And so now you have created a protein-protein interaction that hasn't been there before. And as we've seen for nitrogenase, protein-protein interactions are actually pretty important to build all this diversity that we have in nature. And so this principle, the Tejan lab has used in the past to build discrete oligomers, you know, with no metal in the end, or swapping different metals in, or since you free the metal of its structural role, you can basically use a metal for catalysis, as in this beta-lactamase that Akiv's lab created. And you can also use it to make not discrete oligomers, but periodic polymers, basically infinite assemblies in 2D or 1D. And they can become like micrometer sized and of course crystalline, but still dependent on metal and can fulfill all kinds of mechanical or structural roles. But going back to the discrete oligomers, what has been shown, at least in this example, is that the oligomeric state, so if it's a dimer, a trimer, or tetramer, that depended on the nature of the metal. And this is because the metals, of course, have different preferences of coordination. And the interfaces that basically the metals force this protein into were not strong enough to force a certain oligomeric state, but instead the metal preference is what mattered. And what recently Albert Kakis, who's also a fourth-year PhD student in the Tejan lab, did was he found out that taking that same assembly, basically adding 1 tryptophan, and because this turned out to be not that important, basically using 1 tryptophan, you again get a trimer, and this is with nickel, so that's not very exciting. But if you look a little bit more closely, then you see, yeah, the nickel is still octahedral with 6 cysteines, by the way, interesting binding motif. But also, you see that now the tryptophan is in a T-stack. That's a little bit basically below the metal binding site. And that is important because it turns out that this just gives enough energy to now form trimers. And this is analytical quantification data. So this is basically sedimentation coefficients of this system in solution. And you see that the apoprotein is still, of course, monomeric, but with copper and zinc, it also forms trimers. And it turns out it does form trimers with basically all first-order transition metals, even though, of course, to a differing extent, manganese and iron being considerably harder to bind. But that means it's basically one single mutation that changes from forming tetramers or dimers and to now forming trimers in all of these. And of course, evolutionary, we were interested in, can we drive this a little further, because for iron and for manganese, the binding or the trimerization efficiency wasn't that high. And we used Rosetta and in the end installed a bunch of mutations, which actually not that many, it's only 6, but a couple of mutations which improved hydrogen bonding networks and a little bit of hydrophobicity in the inside. And indeed, even now with manganese, which is probably the hardest to bind of those, we see quantitative trimer formation. And this is at 10 micromolar protein, or 10 micromolar trimer concentration. So it's really quite an achievement. And from an evolutionary perspective, it tells you that basically now this would be a function that is a trimerization upon manganese. So basically, you can think about it as a sensor that senses manganese and then trimerizes. And yeah, we all know that oligomeric state changes are a very important signaling tool in nature. But we can drive this whole series of proteins a little bit further and install a few more electrostatic interactions on the outside of these trimers. And indeed, this now makes this trimer so stable that going from having this purely monomer in the apo state without metal, now we go into a purely trimeric state, even without metal. And that basically means we now pushed it to a point where we can also measure the binding affinities, and they are really high. And actually, they are basically on par with calprotectin, which is one of the proteins in our innate immune defense, and its job is just to sequester metals away from pathogens to limit pathogen growth, right? So from an evolutionary perspective, it's again only a few more changes in the protein sequence, and now we reach something that has a different function. And since now it is also independent of this metal, of course, we can also change now easily the primary coordination sphere of that metal and maybe deseparate it and go towards activity. And I think from the perspective of evolution and metal binding, I think what's important here is to keep in mind how few changes in the structure, in the sequence, can lead to relatively drastic changes in the quaternary structure and therefore also in function. And I think that's important to keep in mind. There's a lot of life at the interface of proteins, which might not be that easy to filter out just from the sequence. Okay, going back to that beautiful work that we've seen before a couple of times, and you all know that of course metal centers are mostly or the real powers and not in them alone, but in their combination. And you see that these edges obviously exemplify the occurrence of these two centers, for example, in the same protein in electron hopping distance. And therefore we asked, okay, can we drive this principle of a metal at the interface a little further and think about it? What if we can put a whole redox active ligand or redox active metal cluster at the interface and bring it into hopping distance with another cofactor and build a new nanomachine, as we call it in this grant from there. And the idea was to use cytochrome CV562 because A, it's a heme, and B, it's basically the favorite building block of the Teja lab. And the idea is we use a bacterial ferredoxin, and we've heard a lot about ferredoxins already in the other talks. And in our perspective, this is of course an exploded view of the two halves, right? What's interesting to me is that the fourth cysteine is coming from the other side, right? So this is basically, if you want, an interfacial fluorine for sulfur cluster, if you just understand the two halves of the ferredoxin as two different domains, so two different proteins, and then that would be an interface. And so the idea is, can we take one piece of this and graft it onto the N-terminus of cytochrome C, the helix, and we try to do that. The fourth cysteine would basically come from a second monomer of that is turned upside down. And then if we do that in silico, then we generate basically a little bit of an interface that then we can improve with Rosetta, putting in some hydrophobics, but also a bunch of H bonds. And it comes down to a distance between the heme and the 4 sulfur cluster, about 10 angstrom, which would be great for electron hopping. And we try to purify it as metal-free protein and reducing conditions. That works pretty well. We did try anaerobic in vitro assembly of the 4 sulfur cluster, which is basically a proven principle using excess iron and excess sulfide. As I think everybody who worked on this, we made the experience that separating the protein from random iron sulfur clusters and oxides, which always crush out of solution, is not that easy. And it turns out that while in UV-Vis, it seems to have a nice peak at around 400 nanometers, what we would expect, which is also reactive to reduction with sodium dithionide. So basically, it disappears upon reduction with sodium dithionide. It has been much harder to show with mass spec or electrochemistry that there actually is a 4 sulfur cluster, and the EPR is also hasn't been positive on that neither. Of course, we would love to crystallize it, and normally that is great if you have these interfacial metal binding sites between existing stable monomer blocks, because they normally crystallize well. In this case, it didn't, probably because it's not a very homogeneous solution going to the in vitro maturation attempt. But since working with Banu and Vic, I've always been kind of itching to try using just anaerobic expression in E. coli and seeing if maybe we get lucky as well, and E. coli just builds the cluster for us. And actually, since now we can slowly go back to lab here in San Diego, I'm trying this week, and we'll see where this is leading us. All right, and with this, I want to thank the whole Tejan lab, and especially also Derek, who worked on this ENIGMA grant before me, and Hannah and Albert, whose work highlighted a little bit, and the rest of the Tejan lab, and of course, the collaboration that has been very fruitful, and Vic and Banu, for whom I hope we can get a crystal structure relatively soon of their protein. That would be nice. Okay, thank you.
Thank you, Julian. Are there any questions? It was an interesting idea with the mutations. So how did you decide on the mutations? Just on the basis of where they're located, or was there any reason for a particular substitution?
In which topic you mean? In the tri site?
Design of the trimer, yeah.
The original.
Yeah, this one. Go back. One more slide. One more slide.
This one?
No, one more. Sorry. There was a list of mutations.
Yeah, forward. These ones.
Yes.
So these ones basically, this was Rosetta design, because we didn't want to disturb this binding site and the tryptophan T stack, and actually it turns out that Rosetta is not able to remodel the tryptophan T stack that we see in the crystal structure. We actually kept the backbone rigid. So this is a rigid body design in Rosetta, and if you do that, it turns out that usually that converges relatively quickly on a number of amino acids, which are much more frequent in the 1000 independent designs that you do. And this is what Albert showed as the frequency over here. And you see that Rosetta really likes these mutations. And so we decided that when it comes out so often, that probably is a good idea. And it turns out that at least for this hydrogen bond network down here, it worked really well. So the distances is basically overlapping with the Rosetta design.
So I am curious because there are lots of tools out there to do mutation analysis, computational mutation analysis. Mm-hmm. So basically, it's just a matter of throwing a couple of residues in positions that you're interested in. But was it particularly the site that was of concern to Rosetta, or of interest to Rosetta modeling? Is that what happened? Or did you not look at other sites, or did it produce all kinds of distributions for other sites as well?
I see what you mean. No, I mean, we basically had Rosetta work on you can basically think about all this interface down here, right? Mm-hmm. So of course the tryptophans and the metal binding site we left alone, but everything else we had in design. It's just that for most of the other sites, within the 1000 designs, it didn't really converge of single solutions. Right? So that basically told us in this case, okay, I mean, we didn't fully trust Rosetta to come up with like the one great design, which I guess it never does actually. And so instead, we decided to basically get inspiration from whatever it suggests. And the way we thought about inspiration was, okay, if it thinks in 900 cases of 1,000 that it's a good idea to put in a lysine at this position, that probably is a very good suggestion. Right, and so that's why we came up with these 6 versus the, I think, 14 different positions, or 24, I forget. I think it was about 20 positions that we allowed him to design.
Got it. Cool, thanks. Any other questions?